is Hungary, right? Well, it takes 24 minutes and then you get your food. Unless I say a little bit extra. So, here we go. This is the first page, and let's start. So if we start from the beginning, and I want to bring it all from big to small, if you think about living organisms, animals, humans, all of us, we consist of many, many cells that hold us together. And in these cells, and around the cells, are proteins. And proteins, we have a lot of proteins in a human being. It's about 20%, that's proteins. And they are the molecular machines of life. And really trying to understand these proteins and how they function and how they come to functional states is where my research centers. So that's what we're going to talk about. But first I want to show you my background, just to kind of see how we move around the world. And that's kind of part of science that it's very international. So I am Swedish, and I got my PhD at Chalmers with Bengt here. Then I moved to America, and I worked with Harry Gray for my postdoc at California Institute of Technology in LA. Then I became a professor in New Orleans at Tulane University. I was there for 5 years. I left 1 year before the hurricane. Then I moved to Houston, Texas, and I was a professor at Rice University for 5 years. And actually, I moved many years before the flooding there now. But in 2008, I moved back to Sweden, to the north of Sweden, a university called Umeå University. You might know about it, it's in the north. And I spent several years there, 7 years. And then now, since 2 years back, I was recruited back to Chalmers in Gothenburg, where I'm a professor, and I'm also chairing one of the divisions in a new biotechnology department. So with that, we go to proteins. So what are proteins? Well, you know we get protein in the food that we eat. The protein is broken down to the building blocks, amino acids, in our bodies. These building blocks, the amino acids that constitute proteins, are taken up in the body. And then within each cell in our body, new proteins are made from those building blocks. And we have thousands of different proteins that perform chemical, mechanical, and physical work in our bodies. That makes us function, in essence. So DNA, the genes that we have in our genome, defines what proteins are made in each cell. That's a central dogma of biology. So the information is stored in DNA, that's translated to RNA, and then transcribed to a peptide chain, or a chain of amino acids, or a protein chain. So proteins are long chains of amino acids, but in order to function, they need to fold into unique compact structures. And that's why I have my snakes with me here. You know Rubik cubes, and this is a Rubik snake, which is really good to illustrate that you have each little piece here is an amino acid. So it's made like a long chain, and in order to function, they need to fold up quickly and smartly into a unique three-dimensional structure, and that's a functional active form. And if they do this in the wrong way, that can lead to aggregation or degradation. And we'll come back to the other ones, I have several. So that's why my research centers to try to understand these type of conformational changes when they go right and wrong. So problems with protein folding can occur because we put the wrong, we have mutations in the DNA that place the wrong amino acid in a particular spot in the protein, and therefore it cannot fold. Or there could be external perturbations. Stress or aging can cause trouble. So, protein folding occurs most of the time without problems and is spontaneous in a cell. So we go from this unfolded state, or after release from a ribosome in vivo, to this folded or native or active structure. So we go from what I call a boiled spaghetti to more advanced yarn ball. And all the information for how to fold is encoded in the linear chain of amino acids. So the question is, how does this work? How does a boiled spaghetti know what structure to go into? That's what we want to understand. And by doing experiments on many different protein model systems, looking at speed and mechanism and energetics and structures involved, we're trying to figure out more about these reactions. We know that folding is fast, and if it were to be a random process, just testing conformational space randomly, it would take forever because there's so many degrees of freedom in a chain like this. So there must be pathways or guiding factors that dictate how proteins fold. And those are the ones we want to figure out, because then we can understand this. So I said most of the time it works. But sometimes we have problems with folding, and that can cause human disease. So if folding goes wrong, that can lead to loss of function. If the protein is not made properly, you lose that function. And this is often the case in cancer, where tumor suppressor proteins are not made properly, for example, p53, and therefore cancer can continue and grow. But then you have the more gain of function or gain of toxic function diseases, and here comes the misfolding diseases, where proteins aggregate into amyloid fibers that cause problems. And here's where I have more than one of these snakes. Because here proteins that should be individually do something come together and form this very long, you should have thousands of these snakes, long fibers that you can see in this picture here. So they aggregate and form these very ordered structures. And that causes trouble. So somehow monomers or single chains assemble into something bigger. It's a little bit like when you boil an egg. When you boil an egg, the proteins in the egg white unfold, and then they aggregate with each other. And when you cool it down, they stuck with each other, so you can't resolve that. And that's why the egg gets hard. It's kind of the same thing with these amyloids. So misfolding or amyloid formation, fiber formation, is the basis of Alzheimer's, Huntington's, Parkinson's, type 2 diabetes, and prion diseases. I think today it's about 50 diseases classified as involving amyloid formation, meaning misfolding and aggregation of proteins. And we'll hear more about this tomorrow, actually, from Chris Dobson. So I want to point out Parkinson's disease, which I will come back to in the end of my talk, because here the name of the protein that forms these bad misfolding amyloid fibers is called alpha-synuclein, and it forms amyloids in brain cells, which is toxic and destroy these cells so they die. It's important to study these misfolding reactions because a lot of people in the world have these diseases. And we think that one way to learn more is to understand correct folding first, and that's the first step to then look into why it goes wrong. So how does protein folding work? Well, what defines how proteins fold? Over the last 20 or so years, there's been many protein folding studies in test tubes in dilute solution. We learned a lot about protein folding mechanisms and speeds from many small systems, which we have characterized. And in fact, I have the world record in fastest folding protein, which I collected when I was in Harry's lab, and I think it still holds. It's about 1 microsecond. But if you think about proteins in living organisms, they're often more complicated than just a single polypeptide chain. So you also need to consider protein-protein interactions when you have oligomeric proteins. So I'm going to illustrate that. If you have a dimer protein, the question is, do these two polypeptides fold first and then assemble? Or could it be they are unfolded and they meet and that triggers fold? That's one question. The other thing is binding of metals to metal binding proteins. So many proteins bind metals, and we heard about that already in several of the talks. So if you have a metal and a protein chain, do they meet before folding or do they meet after folding? And if they meet before folding, maybe they can influence the reaction. And those questions had not been asked before. So my research, when I started my own research group, at Tulane then first, centered on these types of questions to try to understand protein folding on more complicated but maybe more relevant protein systems. And I want to point out, maybe this is no point to point this out now because you know it already, but metal ions are important. So transition metals, iron, copper, zinc, and manganese, they have unique chemical and physical properties that provide functionality to proteins. So if you look in the periodic table, several of the green ones, there are metals that are important for life, and many of them are transition metals. And in fact, 40% of all proteins, or maybe half of all enzymes, need a metal to be functional. So it's a large group of our thousands and thousands of proteins. But we also need to think about that transition metals are toxic in a free form. If you have free copper or free iron, that's toxic. That can cause problems that can damage proteins and cell walls, etc. So we don't want that. So we need to control this in the cell environment. And the problems with metal levels in living organisms can lead to several diseases, neurological cancer, overload, and anemia, depending on if you have too much or too little. Oh my God, I don't have a movie. Ooh. Okay, we'll try this one. So, okay, why is that? I don't know. So, okay, so I showed you on the... Okay, let's look here. Wake up now. Okay, here. This is an example of a protein that has both two chains, or several chains, and metal ions. Hemoglobin, the protein that makes blood red, has 4 polypeptide chains and 4 metals. So I want to show this as an example of a protein that has both metals and several polypeptides. So when we have done studies of several model systems, we found that protein-protein interactions can actually occur before folding for oligomeric proteins. And for metal-binding proteins, the metal can bind to the unfolded state and affect folding. So if we want to understand protein folding of proteins, we need to take these additional factors into account. So let's pass by this. I don't know, this is getting a little bit better. So having said that, we also have to think about that protein folding in a living system is even more complicated. Many times we want to understand how proteins fold in a cell. And then we have to think about the environment in a cell, which is highly crowded with many biomolecules. It's kind of like this picture here that shows a number of different proteins that are packed together. This is how jammed it would be in the cell cytoplasm. And actually they would all wiggle too, but I don't have any movies, so you don't see that. So it's all dynamic and very packed. So it's not this test tube solution that we like to use in our experiments in the lab. So what we're trying to do now in the lab is to understand or ask questions around folding and binding in more in vivo-like conditions. And here we need to take into account how our metals deliver to the right proteins without toxic side effects. And because of this mess or jam in the cell, what unwanted interactions could there be between proteins that could promote disease that are not supposed to happen but might do it because there's so many proteins there? So what we're doing is that we're going more and more from principles to human disease. We looked at the crowded cell environment, how that can in fact, or surprisingly even, affect the shape of folded proteins. That was not expected. But if you change the shape, you can also change the function. We're looking at how copper ions, which is one of the important transition metals that we need in our body, is transported between different proteins to the right target protein. And we found that some of these proteins might play a role in cancer too, that we didn't know about before. And we're looking at amyloid formation in Parkinson's disease. And here we're trying to look at the triggers of disease and if we can find links to other diseases or to bacteria. And I'll come back to this in the end, but first I want to tell you a little bit how we study these reactions in the lab. So first we need protein. And we get protein by using bacteria. And we follow this central dogma of biology. We take the DNA gene for the protein we're interested in, we place it on a plasmid in bacteria, and then we grow the bacteria in a flask. Grows really well, smells. Then you break open the cells and purify out that protein. And this way, you can, in principle, make almost any protein you want, or as long as it can be expressed in bacteria. And also, you can go back and do strategic mutations of any amino acid you want that you want to test. Because you just go back and change the DNA sequence that you have in that plasmid to code for a different amino acid. So then we can make numbers of proteins that we can use to get molecular information about these reactions. Then we do experiments in test tubes. We can study equilibrium experiments. How stable is a protein? What's the shape of a protein? How strong does a metal bind? We can do kinetic, time-resolved experiments. How fast is folding or aggregation? What's rate-determining steps? Or could there be transient intermediates that we can detect? Then we need ways to unfold the proteins, because when we make them in bacteria, they're folded. So then we need to perturb the system so they unfold it, so then we can refold them again. And here we can use heat, as I said, boil an egg, pH changes, or most often chemical denatures that unfold the proteins in controlled ways. We can mimic the in vivo condition using crowding agents that take up a lot of space, or fiddling with salt and pH, or we could add lipids that mimic membranes, for example. And then is the question, how do we detect the reactions? The proteins are in a water-based solutions. You don't see individual molecules here. So therefore, we use a lot of spectroscopic techniques that indirectly see what goes on with the protein. We can use biochemical methods, colorimetry that measures heat, imaging for these long amyloid fibers, and maybe some functional assays too. So we combine a lot of different methods to kind of put a puzzle together. And spectroscopy uses light. We heard about spectroscopy already, but you shine light on a sample, and then you can look at different ways of the light, how that light interacts with the sample. You can look at how much it absorbs. You can look at what light the sample emits, fluorescence. Aromatic amino acids can send out light, and that can tell you about the environment. Or you can twist the light and do what we call circular dichrist to look at the chirality of molecules, and that's a good way to probe secondary structure. So, with that, I want to tell you a few things about misfolding in the context of Parkinson's disease for the last final slides. This is important because more than 10 million people in the world has Parkinson's. It's the second most common neurodegenerative disorder after Alzheimer's, and the number of cases are increasing. And it turns out, if you search around the web, you find this, that every third second, a person in the world is diagnosed with a neurodegenerative disorder. Every third second. So this is a lot of people. And the scary part is that we have no cure. We don't know how to cure. We have some medicine that blocks these diseases for a while, but we have nothing that can cure. So what we want to find out here is really, how do you go from monomers to amyloid fibers? What's the mechanism of assembly? And coming from studies of protein folding, this is very complicated. Protein folding is simple. You have often reversible reactions. It's two-state, highly cooperative, folded and unfolded state, and nothing more. When you come to these assembly processes, you go from nanometer monomers to micrometer fibers. And the monomers assemble to oligomers, and further assembly to larger oligomers, and eventually you get fibers. So it's very heterogeneous, probably. Could be off-pathway species and a lot of things going on. So we want to try to understand, how does it start? What are the intermediate structures? And of these things, what is dangerous? Because remember, this causes disease. And to discover how to modulate, stop, and even reverse these processes, we need mechanistic information. Otherwise, we don't know how to do it. And one key question that we have asked is, what are the roles of other biomolecules? If you think about this crowded environment in a cell, these proteins will always meet some other proteins. Might they affect the reactions, and if so, how? So when we started this, we began actually with small molecules that turned out to be mechanistic tools. So here I want to show you, if I can maybe, I don't know how to use the pointer, but anyway, the blue curve up there, you can just see, here I measure fluorescence as a function of time. So first the blue curve is low, and then it increases. So when the fluorescence or the signal gets high, you have amyloid fibers. So this is the way we probe it. And it takes maybe 24 hours or so, and then it starts to go up. But we found that this small molecule that I show you up here is a 2-pyridine molecule, a flat aromatic a molecule that kind of looks like a dipeptide. It's called FNO75. We found that this molecule would speed up aggregation of alpha-synuclein. So the black curve there that I point to, you see that's shifted to the left. So forms aggregates faster. So then we could modify this chemistry of this molecule and found modifications that would instead result in molecules that inhibited amyloid formation. So in the end, we had a whole toolbox of molecules that would do different things to the protein. And then we could use them to try to isolate and study different species on the pathway. And we found that the mechanism of aggregation for alpha-synuclein goes through various oligomers that you can see as spheres on this image there. And it depends on the secondary structure, which we had to probe by spectroscopy. The secondary structure inside these oligomers, if they will go on to form amyloids or if they will stay as these oligomers, if they were inhibitor or not. So we tried to start to elucidate the pathways here. But then you could also ask, because it was kind of surprising first to us that we found a molecule that would speed up aggregation. So then you can ask, if you can accelerate alpha-synuclein aggregation in vivo, would that give an organism, an animal, Parkinson's? So we decided to test this because we collaborated with people that had mice. So we got to inject this molecule that speed up aggregation in mice. We gave them one injection of this molecule, and we waited 6 months. And then we found several properties of these mice that suggested that they got early Parkinson's disease. I'm not going to give you all the details, but there were cell death in the brain. They had motor dysfunction, and we measured that by putting small stickers on the nose of the mice. And we also looked at metabolites. So they seemed to get Parkinson's. We did some important controls, that's always important. When we add an inhibitory molecule of the same type, nothing happens, which makes sense because the mice are not sick from the beginning. And if we take mice without our protein alpha-synuclein, adding the molecule doesn't do anything to that. So that suggests that this molecule goes into the mice, works on the protein that we studied in vitro in the test tube, and cause aggregation, and that triggers disease. You can ask, why do you want to make mice sick in Parkinson's? Well, this is actually very important because we need new tools to study the earliest events of disease. Most of the current animal models can only look at late stages because you kind of kill off a lot of brain cells and then you see what happens. So new ways to look at the first steps, because that's what we want to cure, so we can cure these diseases early. We also looked at bacteria and bacterial proteins. And this is important because we have about 2 kilos of bacteria in our gut. We have a lot of bacteria, and if you count the numbers, you have more bacteria than human cells, actually. So bacteria will influence us, and bacteria has a lot of proteins in them. So we wanted to see if bacterial proteins would talk to a human protein. And we found, in fact, that some bacterial proteins that we studied that we picked out, they interacted or affected alpha-synuclein amyloid formation. And we could find effects in different directions. We found one protein, CSGC, that can block, that's the blue one there, amyloid formation. Another one that could speed up amyloid formation of alpha-synuclein. So you can go both ways. So then becomes the question, can this have any biological relevance? So here we collaborated with people at Caltech, actually, but totally independent of Harry. He doesn't know this. They have mice without any bacteria in the gut. It's called germ-free mice. And they also express a human alpha-synuclein protein. So it's a Parkinson's disease model. So then we can compare disease in mice with and without bacteria in the gut. And it turns out that the bacteria in the gut facilitate disease. So without bacteria, you're protected. So that kind of says that something in these bacteria, we have lots of different bacteria, and many are really good, but there are some bad bacteria that can maybe trigger these diseases. And we think that maybe proteins from the bacteria somehow come out and escape and find the human protein, and you have a molecular interaction that is the trigger. But then we need to do more to prove that. We also looked at the connection to type 2 diabetes. And this is because type 2 diabetes patients have increased risk to get Parkinson's. We know that from patients, but we don't know why. But alpha-synuclein, although it's a brain protein, it's also there in the pancreas, where diabetes occurs. And if you think about what goes wrong in diabetes, you have a key protein in the pancreas called IDE. It degrades insulin and regulates blood sugar normally. If this protein is not working, you get diabetes. At the same time, you get amyloids of the peptide amylin that form amyloids in the pancreas, and that cause diabetes. So it's kind of two protein systems here that contribute to diabetes. So we wanted to test if there's any cross-reactivity with alpha-synuclein, because they are found in the same cells. And what we found is that this IDE protein is a very good inhibitor of alpha-synuclein amyloid formation when it's there. We also found that this amylin amyloid that causes diabetes can speed up aggregation of alpha-synuclein. So it goes faster. You shift the curve. So it's really two ways here that you can see a connection between these two diseases. That this IDE protein is gone in diabetes, and therefore alpha-synuclein can start to aggregate much easier. And the seeds of these other amyloid protein that's there in diabetes speed up alpha-synuclein aggregation. Of course, now we need to see what happens in vivo, but you need to look at these molecular interactions in a test tube first and see what happens. Finally, I want to mention membranes, because membranes are all over the place. You saw on my very first slide that there were millions and millions of cells, and cells are lipid membranes. That's how they hold together. And within each cell, there are compartments that have lipids to confine them. So lipid membranes keep cells and compartments together. Can that affect alpha-synuclein aggregation? It's an important question because it's been proposed that alpha-synuclein binds and transports vesicles in brain cells. We actually don't really know the normal function of alpha-synuclein, which is kind of interesting. But an idea is that it binds to lipid vesicles and moves them around and maybe send them out with signals in. So we decided to test interactions of alpha-synuclein with different kinds of lipids or lipid vesicles. And here I show you two lipids that have the same charge. They're negatively charged both, but they have slightly different chemistry in the head groups. And we found that this chemistry in the head group of the lipids, which is what sticks out on the surface, matters for binding of synuclein. So depending on the lipids we used, we got different binding modes of the protein to the surface. And this also affected how the protein aggregated. So we found that in most cases, membranes can accelerate amyloid formation, but only in a certain window of concentrations, because you also need some free protein, and you can't have too much bound, because then you can inhibit instead. So there was a lot of constraints here. So we think it's very important to continue and study lipid interactions. We have a lot of different lipids in the membranes we have in the body. So there's a lot of different lipids to look into, and combinations of those. But it's important for the connection of function and dysfunction. Where does aggregation start? Maybe membranes are there in the normal function, but then they trigger amyloid formation. So with that, I want to conclude and give you some take-home messages. Proteins are essential. Most proteins fold to function, but if they misbehave, they can cause disease. So we have correct folding, and we could have misfolding and aggregation. Yeah, I look at you instead. Close your eyes. Misfolding and amyloid diseases increase in the world. That's true. The major risk factor for these diseases is age, and the population is getting older. So we're just going to see more and more of this in the future. And people would predict that this can overwhelm the healthcare system if we don't do anything. In fact, already today, it's a larger risk for elderly to die from amyloid disorders than from cancer. I think this is kind of scary. Yeah. We need more molecular research on why misfolding starts, how it spreads, and the biological consequences to learn how to counteract. And we also need to take into account the complexity of the cells, such as other proteins, metal ions, and membranes. But we need to do this in a controlled way so we actually can interpret the results that we get. With that, I also want to say that research is fun. It should be fun, and I hope you will have fun with research in your future. Here I want to show you the last slide. The students in my group, the people that actually do the work now. This is my research group at Chalmers. About half of these people work on alpha-synuclein and amyloids. The other half work on copper transport proteins, and actually 2 students work on diabetes too. With that, I want to say, well, I should point out one student here. Now I'm going to test something. No. Okay. Stop. One more thing. I wanted to try one thing. My idea was to point to a certain person on my last slide, but I couldn't. But one of my students, or senior researchers, he is from here. He graduated from this university, and I wanted to point him out, Istvan Horvath. And he was so happy and told me everything about it before I came. But thank you for listening.